Hello everyone, and thank you for joining us for today's online workshop organized in collaboration with Surface Measurement Systems, Johannes Gutenberg-Universität Mainz, and the Max-Planck-Institut for Chemicals. We're really thankful to have our two guest speakers, Moritz, Rosch and Johannes Blecher, with us today to share their results. So straight off the bat, I'd like to offer a huge thank you to them. Before we continue with today's presentation on sorption analysis in pharmaceuticals formulation, I just wanted to remind our audience of the ways they can ask questions at the end of each presentation. To ask questions, you can either submit them in writing in the questions panel, or you can raise your hand at the end of each presentation and I will let you know when you can unmute your microphone, and you can ask the question yourself over your mic if you would like to. So once again, you can submit them in writing, or you can raise your hand and I will, you'll be unmuted when and given the opportunity to ask your question. We will have a Q&A section at the end of each presentation, so please do submit your questions as you are listening to the presentations to make sure you're getting your questions in there. So before we continue with the main part of the workshop, I'm just going to hand over to Thomas Schmidt, sales manager for the DACH region for Surface Measurement Systems, and he'll just give you a quick introduction into Surface Measurement Systems, our instruments and our science, just to give you a quick grounding of knowledge to inform you throughout the rest of today's workshop. So I'll pass over to Thomas now.
Okay, thank you, John. Good afternoon, everyone. I thought it may be useful that as some people we don't know some people in the audience, that we do a short introduction. It will not be long before we start with the application talk, introduction about surface measurement systems and how we can help and support you. I hope you can see my screen in big size.
It's all good, Thomas. Yes.
Okay, fine. Yeah. So surface measurement systems, what are we doing? We develop and engineers techniques and instrumentation for physical-chemical characterization of complex, most solids, can be geels also, but most of materials we are characterizing with DVS and IGC are solids. And we have become world leaders in dynamic vapor absorption, known as DVS, and inverse gas chromatography, and that providing very good scientific support and also technical support to all our customers, and that nearly 30 years now. Yeah, our expertise and our strengths are, of course, the instruments, all right, that is basic, but also our application scientists and product experts. Yeah, we are providing you over the whole life a direct support and a practical R&D support, and we work closely with customer. We like to work closely with customer to follow what they are doing, to improve our system, etc. And of course, important is a dedicated team of service engineers who are there to assist you, to support you with visit and service contracts. Last but not least, we provide a series of symposium, webinars, or workshop like this to present some scientific work and to have a good meeting and conversation with you. So don't hesitate to ask your question over the chat or after the talk. We have offices in North America, India, and in the UK. Representatives all over the world, and we will open shortly or in the next time an office also in Central Europe, in Germany, so in the Germanic region, exactly for in the area where most of people are which are in the meeting here. Yeah, so we're world leaders in dynamic vapor absorption instrumentation, and the company was established in 1990. That was the beginning, established by Darrell Williams. He was born in Australia. He moved to London in 1986. And yeah, he is multifunction reader in particle science. He is a professor at the Imperial College. He's director of development in a place called Discovery Space at Imperial College London, and director at Surface Measurement Systems. He has published a lot of paper, over 70 papers, and 5-6 patents in the area of vapor absorption technologies. And the first customer was Pfizer. It was a pharmaceutical customer, 1991. And we delivered the first dynamic vapor system as analytical tool, and the first instrument available on the market at this time in 1992. So what are our products today? We're experts in dynamic vapor absorption instruments, which allows you to measure the uptake and loss of moisture or organic vapors, or also mixtures. We'll see more later in this workshop. And surface energy analyzer by IGC. And IGC allows to characterize surface properties, but also bulk properties, especially of powders and particles and fibers, where it is very, very strong compared to other techniques available to measure surface energy. The vapor pressure analyzer measures the vapor pressures of liquids and solids under different conditions. Last but not least, the DVS vacuum instrument, dedicated, I would say, for more microporous materials, porous materials, because it allows you to go to very low partial pressure. So that is the range of DVS you have today. First, on the left side top, the DVS intrinsic for water absorption between 20 and 40 degrees. In the middle top, it's a DVS adventure. It covers a broader temperature range between 5 and 85 degrees. And it has also some options as camera or in situ Raman spectroscopy to better see some transitions even over that microscope over Raman. You have the DVS resolution top right side. The difference is the adventure is the capabilities of vapors. It can do one vapor or two vapors or in a mixture, and there are two configurations: standard water with organic and the advanced system organic with organic. And sorry, there's a new thing, we can up to 3 vapors with water with a DVS resolution. On the bottom side, you will see the DVS Endeavor. It has the capabilities either of the DVS Adventure or the DVS Resolution. The big change in the system is you can measure up to 5 samples fully in parallel, means you will measure the full kinetic of 5 samples because of 5 balances in that system. And you get the same for discovery. At this time, water only, but we will expand the capabilities to vapor next year. And it allows to measure 2 samples at the same time. Sometimes interesting when you want to do a second measurement, you can do it in the same time in that system. And last but not least, left side bottom, the DVS vacuum for microporous materials. It has also that vacuum system, of course, and a preheater which goes up to 400 degrees. As I said, mostly drying experiments I would see in the pharmaceutical industry, or if you have porous materials, especially microporous material, you would like to study. What can we do generally with DVS? Just to pick up, I would say some possibilities you have. Of course, you measure how your material, how your sample interacts either with moisture or solvents, and then at different temperature and partial pressures. You can measure the stability, performance, and processing. You can see if the sample is stable with changing humidities or changing partial pressure, and see the effect of, for example, humidity, if it is reversible or irreversible. If you get, for example, polymorphism, in function of relative humidity. You can measure the moisture content and moisture isotherm between 0 and 98%. That may change at high temperature, but we could cover the full humidity range, means from dry 0.0% up to 98%. Identify the heterogeneity of sample batch, or identify variance of an homogeneity within one sample. DVS, of course, you can follow the kinetics, measure the transport properties, means measure your sample at 1 point per second. You can do some measurement related to energy, measure how strongly the moisture or other vapor is bound to the material, to the surface or the bulk of the material. 8, you can identify phase transition, glass transition, recrystallization, polymorphism in function of humidity or other vapor pressure and temperature. And that's, I would say, a big difference to DST, for example, where the humidity is constant. Hydration and solvate formates, drying analysis, diffusion and activation energy, that has to do, of course, with kinetics, the heat of absorption, and of course, last but not least, I would say I think you do all that, measure moisture uptake and content. What amount of vapor is taken and released. And here, that's the other guy, that's the IGC-SEA. IGC-SEA, you measure surface energy, which is important for the formulation design, to measure and to predict the processing performance, and also the quality of your formulation, C cohesion and adhesion properties. What is also a main application of IGC in the pharmaceutical industry is to measure the surface energy heterogeneity of your sample, and especially of the surface. Yeah, you may have some changes of the surface you will not detect with other systems as calorimetry or DVS, when it's very fine and very sensitive changes, but you can detect it with IGC. And that is something where IGC and gas chromatography is very sensitive. So here you have a range, not sure that go through, but of properties you can measure with IGC-SEA. Perhaps I pick up 3 properties. We have surface energies. BET, specific surface area, is very good to measure with IGC, especially if the surface area and the specific surface area is low. It is very sensitive, and you can measure it at room temperature. You can even change conditions, humidity, to measure surface area at, I would say, environment conditions, and not at low temperature, 0% humidities, but for example, 25° and 40% humidity. And the last I pick up is heterogeneity mapping, surface energy heterogeneity. For software, something important where, I would say you run your system, you analyze data. It's an in-house development. So we are really experts also in software development. Yeah, people who knows what measurements are done to do a comprehensive, intuitive, user-friendly software. And that's the feedback we get from most of the customers. That is very user-friendly. And it's based on the latest operating system, Microsoft. So here, last picture to say hello. That's a little team of some people of the team worldwide. You see here also some people from production. We are here to support you, and that's the message of that picture. If you have any question, you can call us at any time. It's our job. That's what we like to do. Okay, I thank you very much, and I would pass to the first speaker, And I think it's Johannes Blecher who will have the first talk. I pass to John, who gives the transition. Thank you very much.
Thank you very much for that, Thomas. And yes, quite correctly, our next speaker is Moritz Rosch from Johannes Gutenberg-Universität Mainz and the Max-Planck-Institut for Chemicals. And he'll be discussing DVS analysis of different citric acid excipients for pharmaceutical formulation of effervescent tablets. Just a quick reminder to our audience, you can submit questions for the end of this presentation's Q&A by submitting them in writing in the questions tab, or by raising your hand at the end of the presentation, and I will hand over mic control to you. So without further ado, I will hand over to Moritz.
All right, so welcome everybody. My name is Moritz Rosch, and I'm a PhD student at Johannes Gutenberg-Universität and Max-Planck-Institut for Chemistry in Mainz. My presentation today is about dynamic vapor sorption analysis of different citric acid excipients for pharmaceutical formulation of effervescent tablets. Specifically, we are going to take a look at hydrogen generating effervescent tablets. And why hydrogen generating tablets? You might ask yourselves, well, I will tell you about the effects of hydrogen as a medical gas, and afterwards show some of the stability investigations that we did with dynamic vapor sorption during the formulation development. So hydrogen as a medical gas has antioxidant and anti-inflammatory properties. The main mechanism is the reduction of reactive oxygen species and reactive nitrogen species. Here are some examples. For example, the superoxide radical, hydroxyl radical, and peroxynitrile anion. And besides radical scavenging, there are different mechanisms who are not yet discovered and discussed, like the activation of the body's antioxidant own system via the factor NRF2 is currently under investigation. Yeah, moreover, there are animal and human studies regarding different topics. For example, chronic fatigue is a topic there, and COVID-19, obviously, especially with long COVID symptoms. But also Alzheimer, Parkinson, diabetes are discussed, and also performance enhancement of athletes is a topic that is investigated for hydrogen. Hydrogen as a medical gas is applied either via inhalation, then you have 1 to 4% gas. It can also be used as a liquid, then it's called hydrogen enriched water, and it can also be injected. For gas and injectables, you always have to have qualified personnel to use it, and the hydrogen enriched water can also lose the hydrogen when you purchase it directly. So our aim was to that patients can prepare the hydrogen enriched water independently of qualified personnel and also freshly, so that it doesn't lose its hydrogen. Here you see some established products in Japan. There is one hydrogen enriched water, and the second is electrolysis hydrogen water generator, and there are even water taps who produce hydrogen enriched water. Yeah, but as I said, our approach was to have a tablet to prepare this hydrogen enriched water. Contrary to an conventional effervescent tablets, you have a different effervescent reaction. For the usual effervescent tablets, sodium bicarbonate reacts with an organic acid and forms salt of this organic acid and CO2 gas. And in our case, we use metal magnesium, and we also have an organic acid as a reactant, and the magnesium is oxidized and reduces the protons of the acid, basically. So we form our intended hydrogen gas. Effervescent tablets are usually really moisture labile, because technically effervescent agents constitute an incompatible mixture within the tablet. And humidity also triggers these effervescent reactions that I talked about unintendedly. And during manufacturing, it also can cause sticking if it's very humid. For example, you see it on the right, sticking occurring during compaction. And moreover, this is all worsened by the fact that citric acid is hygroscopic. So yeah, as you can imagine, this is a really problematic formulation to manufacture and store. So we use the dynamic vapor sorption to evaluate different citric acid products and avoid issues with sticking and stability. We had a citric acid anhydrous powder, citric acid granular, and also a citric acid granular that was coated with sodium citrate. For our experimental setup, we chose to start at 0% relative humidity and go up to 90% relative humidity in 5% intervals. And this is done at 5% intervals at 25 degrees. And since we want to measure the resistance toward humidity and our product reacts when it's exposed to high humidity, it will be irreversibly destroyed once it takes up too much moisture. So we didn't investigate the desorption process too closely. So we had 3 steps of desorption. Just from 90%, we went to 50% relative humidity and then to 0% relative humidity. Since we expected the sample to take up a lot of water, we chose not so strict equilibrium criterion, and we chose also a long time frame of 600 minutes if the equilibrium criterion cannot be reached, so that the sample has much time to possibly reach its equilibrium. So now we're going to take a look at the powder-based blend first. What you can see here is the experiment that I just described, and what we saw was that we have no meaningful water sorption measured during the first stages of 0 to 55% relative humidity. And at 60% relative humidity already, the sample increased 1% in weight already. Then during the 65% relative humidity stage, high amount of water sorption took place. The sample increased its total weight by roughly 11%, and at an average mass increase per minute of 0.016%. The total mass change was roughly 49%, which is quite a lot for water sorption. So next would be the granule. Here you already observe a difference because there was no meaningful water sorption until the stage of 60%, in contrast to the powder, which already took up water at 60%. And then during the 65% stage, the granule also took up some water, but in contrast to the powder, it was only roughly 6%, so roughly half of the amount of weight gain. And also the mass gain per time, as you can see with the average DMDT, was slower. So the mass gain was slower and the total mass gain was less. The total mass gain after all stages was 45%, so there is no big difference there. And then we come to the coated granule-based blend. Similar to the granule, we have no meaningful water sorption until 60% partial pressure, and also pretty similar during the 65% partial pressure stage. Roughly 7% weight increase took place, and also the rate was roughly similar to the granule that we observed before. Total mass change was relatively the same for all samples, in this case 46%. So what did we get from these two results? The total mass change was relatively similar for all samples. Differences occurred in the starting point of the sorption process. For the powder-based blends, it was at 55% relative humidity already, and for the granules at 60%. And also for the sorption rate, there were differences, the powder being the fastest, showing the fastest sorption, and the granules also relatively similar and slower. One explanation for this would be that the powder has, due to its smaller particle size, has a larger surface area, and thus the sorption process started earlier and also was faster compared to the granular excipients. And yeah, regarding the granular excipients, there were no noticeable differences. One possible explanation that we concluded was that the coating, that the moisture protective coating of the coated excipient, of the coated granulate, was probably negated, because in the mixture there was also mild adipic acid of very fine particles, which would have represented a large part of the accessible surface, and this is in comparison a relatively hydrophobic excipient. So for the granulates, there was not a big difference. And yeah, so based on this, we could decide on the excipient and continued with the coated excipient. We also wanted to take a look at the reaction a little bit more. So we did a 2-cycle screening experiment as well, with 2 sorption and desorption processes from 0 to 90% in 10% intervals to keep it a little bit short. And also the equilibrium criteria were the same as shown before. So here you see that the first cycle is comparable to the previously shown experiments, including also the incomplete desorption phase. And the second cycle is different. It shows an earlier mass increase and a lower maximal mass. So this clearly indicates that a chemical change took place here. Otherwise, we would observe 2 identical sorption and desorption cycles. And possible explanation for the data that we see here is that after the reaction and partial dissolution and recrystallization at the interparticulate interfaces took place, which resulted in a reduced total surface area that was available for water sorption in the second cycle. So based on this DVS results, we also conducted a bike stability study of unpacked tablets. We did this in a constant climate chamber. And from what we learned from the DVS results, we expected the tablets to be stable at 25 degrees and 60% relative humidity. which was relatively true over a moderate period of time. So after 14 days, we observed about 1.3% of weight gain, and after 8 weeks, we had 2% of weight gain. And we also took a look at the hydrogen generation speed and the hydrogen generation amount of the tablets. There you can see small differences. So the initial non-stored samples, the T0 samples, show a quick and complete hydrogen generation. And over time, there is a decrease in the hydrogen generation speed and also in the amount of hydrogen that is generated. So after 8 weeks, it is significantly decreased in this case. So based on the DVS results, we gained important knowledge of our formulation, which enabled us to predict its stability during manufacturing and storage. So this was why DVS was very useful for this project. So thanks to my working groups of MPIC, Max-Planck-Institut for Chemistry, AG Lucas, and my group from university, Johannes Gutenberg-Universität, AG Langgut, and thank you for your attention. Thanks.
Thank you very much, Moritz. We'll just give the audience a minute or two just to submit any questions they may have. So please, just a quick reminder to our audience, you can raise your hand or you can submit the questions in the questions panel. So please submit any questions you have for Moritz now, please. A question here, Moritz, and they ask, what coatings are usually used for the granulates?
The granulates themselves are not coated after processing. The citric acid is available in different grades. So this is an available excipient that is coated with monosodium citrate, 3% of the weight, basically, of the excipient.
Great, thank you.
And it's less hygroscopic than the citric acid. So they have a monosodium citrate coating.
Yeah, we can't see any other questions for now. So just a reminder, if you do have any questions for Moritz, please do enter them in the questions panel or the chat, and we'll make sure he comes back to you. But for now, we will move on to our next presentation for the day. So a massive thank you again to you, Moritz, for giving a presentation with us here today.
Thanks.
Our next speaker will be exploring the evaluation of moisture protection in solid dosage forms, and this will be presented on by Johannes Blecher from the Johannes Gutenberg-Universität Mainz. So without further ado, I would like to thank Johannes for being with us today and pass over to him to make his presentation.
Yes, so thank you, Moritz, for this interesting presentation on hydrogen forming tablets and DVS application. And I would like also to welcome all of you to our webinar. The topic of my webinar will be the evaluation of the moisture protection in solid dosage forms. So first, I want to give you a quick outline. I will start and give you a brief overview why moisture protection is an important topic in pharmaceutics. And then I will show you 3 applications on how to assess this. The first one is the water diffusivity in polymer films that are used for coating. The second application will be a sorption and desorption on uncoated and coated tablets. And the third application will be the determination of water vapor permeability. So first, let's start why moisture protection is important in pharmaceutics. On the one hand, there are APIs, like for example, acetylic salicylic acid, that are susceptible to hydrolysis. And for this reason, this API would lose its irreversible inhibition function of its target and lose its efficacy. On the other hand, there can also happen some physical chemical changes in drug formulation, which, for example, can change the drug release. Here in the middle graph, we can see some dissolution results from erythromycin tablets, and they were tested immediately after manufacturing and after 40 days of storage at different conditions. And as we can see clearly from the unstored tablets, the dissolution dramatically decreases when, for example, the tablets are stored only in paper bags or in plastic containers, and only glass show the more or less adequate storing capability. Second example is the disintegration time, which can also be changed due to moisture at different after storing. And here in this example, the two formulations were stored up to 70 days, and as we can see, also the disintegration time increased dramatically in these two formulations. So as a result, this could change the bioavailability and so also the plasma concentration time profiles from these formulations. And two ways to avoid these changes in our formulations are, on the one side, to coat our formulations with functional polymers, and on the other side, use proper storage conditions like primary packaging materials, as we can see in the middle example here. So let's start with the water diffusivity in polymer films that are used for coating of, for example, tablets. The polymers used in our experiment were the Eudragit L55 and HB50. These two type of polymers are usually used as enteric coating, but of course an additional benefit of them would be the protection against moisture. So what I did was I prepared some thin polymer films by film casting from organic solution, and then put these polymer film fragments into the DVS apparatus and run cycles like displayed here in this image. So we dried the polymer film first to get the zero mass, and then increase the relative humidity to, for example, 20% or up to 90% even. And from this, we could calculate the water diffusivity. And this can be done in the following way. According to Crank and Park, we have the following formula, which is valid for as long as MT divided by M infinity is smaller than 0.4. In this case, MT is the mass of our sample at time point T, and M infinity is the mass of our sample at equilibrium at a given relative humidity. D, the small d here, is the thickness of our polymer film. The large D is, or capital D is, the water diffusivity. And so if we plot MT divided by M infinity against the square root of T divided by the thickness, we should get a linear plot of our sample. This can look, for example, like this here. And from the initial part of our graph, from the slope, we then could calculate our diffusion coefficients. And as we can see in both polymers, they are quite low with 1.5 times 10 to the minus 8. For example, at 80% for the Eudraget L55 polymer. And with a higher diffusion coefficient, of course, the permeability through this film would increase. So with low values, we are good to protect our dosage form. This can also play an important role, for example, in solid dispersions or solid amorphous dispersions, because when water can act as plasticizer in polymer films, so if we would have a high diffusion coefficient, it would mean that more water could be taken up into the polymer and change the glass transition temperature, and accordingly affect the stability of solid dispersion. But that's for the assessment of the water diffusivity as first example. The second example was the water sorption and desorption we did on uncoated and coated tablets. In our case, we used two brands that are on the German market, the aspirin and the ASS Ratiopharm tablets. In this case, they are not coated, and what we can see is that we have for the aspirin tablets, for example, absolute mass increase of around 3.5 milligrams, and for the Ratiopharm brand, even up to 4 milligrams. From these plots, we can now take the percent weight gain at equilibrium at every relative humidity stage and plot the sorption and desorption isotherms afterwards, when we plot the weight gain against the percent relative humidity. And according to the European Pharmacopoeia in chapter 5.11, we can classify now our material or product as hygroscopic or non-hygroscopic. And this chapter describes that when the weight gain at 80% relative humidity, so at this point here, for example, is above 2%, but lower than 15%, our material is hygroscopic. If it's above 15%, then our material is very hygroscopic. And when it's below the 2%, but above 0.2%, our material is slightly hygroscopic. So when now looking at our two tablet formulations, we can see that the ASS Ratiopharm tablets are hygroscopic, whereas the aspirin tablets are only slightly hygroscopic. These experiments were also performed with the coated formulations of both brands. So we have on the left-hand side the aspirin protect tablets, and now we can see already a difference to the uncoated tablet. The mass increase is only up to 0.8 milligrams. In the uncoated version, it was about 3.5 milligrams. And also up to a relative humidity of 60 to 70%, we almost have no increase in the mass, and then we have the increase in mass here above this value. Something similar was seen for the coated ASS Ratiopharm tablets. Up to a relative humidity of 70%, we have only a slightly increase in mass, but then a significant increase. But this increase is also lower than for the uncoated formulation. So again, when plotting the sorption, desorption isotherms, we now can see that both values at 80% are much closer to the 0.2 in both formulations. So these formulations are only slightly hygroscopic, and are expected to be much more stable when stored at a given condition. Okay, so at last we can also protect, of course, our formulation with the right choice of packaging material. So we assess the water vapor permeability of some packaging materials. This can be done using a paint-type diffusion cell, which consists of a small lower cup where there can be put a drying agent inside. For example, we use the silica gel. And between two O-rings, we can put then our test specimen and close this with the cell upper lid. To now calculate the permeability, we first need to know the moisture vapor transition rate, which is the change in mass over the change of time. From this, we can calculate the vapor flux, which is the moisture vapor transition rate divided by the surface area of the paint-type diffusion cell, which is 15.54 square millimeter. And then the permeability can be calculated by dividing the vapor flux by the difference in relative humidity from inside the cell and the environment. So, as was also done in literature, we investigated paper bag as a material, which is sometimes used in public pharmacies when, for example, powders are prepared for specific patients. So we dried the sample again for 2 hours at 0% relative humidity, and then increase the humidity to, for example, 90% in this picture, but also to 30 or 60% at 25 degrees. So we get a graph like this, and again, from the initial part, from the slope, we can then calculate the mean vapor transition rate. So we can see at 30, 60, and 90%, we have an increase in the moisture vapor transition rate, which is a higher slope, the blue line here. From this, we calculated the water vapor flux, and in the end resulted in the water vapor permeability of this paper material being between 2.6 to 4.8. We also investigated the permeability on aluminum blister sealings, which is typically used in pharmaceutical. And what we see here is that we have a much lower flux of the water vapor, resulting also in a lower water vapor permeability, being around 10 times less than in the paper bag situation. Last, we also applied this method to a Eudragit L155 film. So this Eudragit is again a coating material. So if we have a casted film, we can also test a coating formulation in this experiment. And what we saw here is that the water vapor permeability is even lower than in the aluminum sealing seen right before. And with this last slide, I would like to thank you for your attention, and I'm now happy to receive your question. I hope I could give you some insights to the applications of the DVS in pharmaceutics.
Thank you very much, Johannes. And yes, a reminder to our audience to now submit your questions for Johannes in case if you have any queries about any of the topics discussed here. So we'll give you a few minutes just to submit your questions. We have a question here, Johannes, and they ask, is the total water uptake relevant to any other properties of different citric acid samples during, oh, this may have been for our last presentation actually, so we'll get that to Moritz. Apologies. Yes, we have a question here, so let's pull this up. They ask, how did you measure the thickness of your coating?
Okay. The thickness of the coating was not, I mean, this was not an applied coating to a formulation, but we casted some polymer films on a glass plate, and then we could simply measure the thickness using a caliper, and it was about 750 micrometers, I think, if I remember correctly.
Thank you. So while we're awaiting any other questions for Johannes, Moritz, would you be able to just to give some insight on this question? Is the total water uptake relevant to any other properties of different citric acid samples during the application?
Yeah, well, for us, as I pointed out, it was most important for the manufacturing and the storage, but it affects many properties actually of the products in between and the tablets as well. So maybe you remember the last slide where I showed the hydrogen generation of the tablets. So even the 8-week stored tablets that were stored in the climate chambers, for example, they took only up 2% of water, but this made a really big impact on their disintegration speed, for example. And yeah, also the hydrogen generation amount, because the reaction took place there. Yeah, but to sum it up, it was most important for manufacturing and storage stability, I would say. Yes.
Brilliant.
I hope this answers your question. Otherwise, feel free to specify a little bit more.
Thank you very much. We have another question for Johannes here, and they ask, how did you choose the length of the measurement when using the pain cell?
Sorry, could you repeat the question, please?
Of course. How did you choose the length of the measurement when using the pain cell?
Okay, I choose a dm by dt criteria of 0.005. So if this dm by dt value was achieved, then the measurement stopped and equilibrium was assumed.
Brilliant, thank you. Question here. To determine whether the sample is hydrogroscopic, do we need to completely dry the sample and then measure the full sorption isotherm? Or if the sample will lose the structure at 0% relative humidity, can we start from 10% relative humidity, perhaps?
I think we don't need to measure the full cycle. The pharmacopoeia specifies that it has to be measured at 80%. I would have to check the exact prescription there, but I think from 10% could be fine too.
Thank you. And we have Jacob Domeni has asked to raise his hand. So Jacob, we have unmuted you if you would like to ask your question.
Hello, do you hear me?
Yes.
Perfect. Hello, I'm Jacob from Czech Republic. I was asking how long you set the length of the measurement for the pain cell, because I thought that using this pain cell there's no possibility to choose DMDT, because if you have a silica gel inside, it will still take more and more moisture, so there will be no established DMDT. So therefore, I was asked, can you explain it? How did you choose the set it time, or there was really DMDT settings?
No, there was really the DMDT setting. I mean, of course, you could play with this and say it more strict or less strict, but in our case, then at the 0.005% per minute was fine to establish then the equilibrium.
Okay, thank you very much.
Welcome.
Thank you, Jacob. Okay, I think that brings us to the end of the questions on this presentation. So thank you again, Johannes, very much for being with us today to share your findings with us. Moving on now to our final presentation of the day. The topic being discussed here is the effect of temperature and moisture on sorption properties of microcrystalline cellulose. And this is being discussed by our very own Meishan Gao from Surface Measurement Systems. So once again, remember, we'll have a Q&A at the end of Meishan's session. So please do raise your hand or submit your question in writing if you'd like to ask a question. So handing over to you now, Meishan.
Thank you, John. I'm just going to share my screen, so I hope you can see it now. Okay, that's great. And thank you very much for the nice presentation in the from the previous two speakers. It's really nice application of DVS technique. And in my presentation, I will just give a more general introduction of the two techniques, both the DVS and the IGC technique for the applications. And here I will just focus on the data with the macrocrystalline cellulose. So here just the outline of my presentation. So first we will have a brief introduction about the sorption techniques, because both DVS and the IGC are the sorption techniques mechanism. And we will have a separate part. So one section is for the DVS, a general introduction for DVS, and we'll focus on the temperature effect for the sorption properties of the microcrystalline cellulose sample. And for the IGC part, we will also have an introduction for the technique and the case study related to the effect of the humidity on the surface area of the MCC sample. So for the sorption technique, with the characterization of the solid material, we can either use energy or heat as a probe. And in case of the sorption technique, we will use molecule as a probe to interact with the solid sample in the bottom here. So by studying the kinetics of this sorption and the desorption, we can then determine some of the property of this solid material at a certain conditions at a combination of the temperature and relative humidity. So in case of the interaction of the probe molecule with the solid material, it will be either absorbed on the surface of this material or absorbed into the bulk of this material. For the absorption on the surface, it will be either by physisorption or chemisorption, depending on the bond is forming for the covalent bond or just loosely bound on the surface. So from the absorption interaction, we can then determine lots of surface-related property. So for example, the surface area or the surface energy of this material at certain conditions. And in case of the absorption, it could be either by bulk sorption or the absorption into the lattice structure. So the bulk sorption will more related to the vapor-induced phase change happening into the bulk of the material. So we can determine the glass transition temperature and humidity for the sample. Or we can also determine, in case of the previous presentation, for the permeability or the diffusion of the water vapor through the thin film sample or through the particle or the powder sample. And the last kind of interaction is more related to the hydrate formation or solvate formation in case of the organic vapors. So we can study the kinetics of this hydrate formation process or the structure of this hydrate. So first we will focus on the dynamic vapor sorption technique. Just a brief introduction. DVS is a gravimetric method that we measure the mass change of the sample at different concentrations of moisture or organic vapors. So we based on a very accurate balance in the system with the resolution of 0.1 microgram. And this will allow us to use very small amount of sample for the analysis, typically just around 10 milligram, as long as the water uptake of the sample has a good uptake. And because we are using small amount of the sample and the resolution of the balance is very sensitive, we can achieve a much faster equilibrium compared to the static method. So a full sorption desorption experiment could be happen just within 3 or 2 days. And the measurement of the DVS technique can be vacuum condition or the ambient temperature pressure. So we can simulate the conditions during the application of this product, and we can get more practical properties for the storage during the storage or the processing of this material. So here's a schematic of one of our DVS instruments, the DVS resolution system. So this system can perform the sorption of both water or the organic vapors. So in the middle of this system, we have a macro balance, which is connected to both the sample pan and the reference pan. And we will put the sample pan here, and the change of mass will be recorded by the macro balance. So the carrier gas for the system will be nitrogen or dry air. And as you can see, there are 2 bottles at the back of the system. So on the left-hand side is for water, and on the right-hand side is for the organic vapors. So the carrier gas, the mass flow controllers will control the flow of the carrier gas with the saturated vapor in a ratio to reach a certain level of humidity. And the sensor here will detect the concentration of this mixture to make sure the actual generation of the vapor is matching with the target values. So this mixture will flow to both the sample chamber and the reference chamber at a constant rate, and the sample inside this chamber will pick up the moisture. Then the mass will be recorded by the balance. So the whole setup here will be set in an incubator with the temperature range from 5 to 85 degrees, which means we can perform experiments with a very wide temperature range from 5 to 85 degrees. And the DVS technique could also be integrated with some other accessories. For example, we can put a camera under the sample chamber here, and then we can use a transparent quartz pen and take the photo images from the bottom of this pen. So we can record any change in the nature, for example, the swelling or the shrinking behavior of the cellulose sample, or the recrystallization of this crystalline sample at different humidities, and the conditions will be recorded in the ledger here. So the video function is a very powerful tool to record any change in the nature. And another accessory to be integrated with DVS technique is we can put the Raman spectroscometer with the DVS, and we can collect the Raman spectra at the end of each humidity stage when it's reaching equilibrium. So for example, here we have the amorphous lactose sample, and we perform 2 cycles of water sorption and desorption experiments with this amorphous lactose sample. So the right line here is the change of mass for the sample. So below 60% RH here, this amorphous lactose sample has a very good uptake. It's reaching equilibrium and then jump to the next stage. And at 60% RH, you will notice there is a significant decrease in the mass for the sample, and that's because of the amorphous to the crystalline phase change, because the crystalline material has smaller surface area as well as less sorption capacity. And this kind of phase change is an irreversible phase change that it doesn't happen in the second cycle. So that's just some observations from the DVS data. And we also use the Raman spec to collect the spectra. Here are the data showing. So at 0 and 40% RH, it's just some broad peaks in the Raman spec. We're just confirming with the amorphous state of the material. And above 60% RH, there are some new peaks showing up here. And these new peaks is because of the crystalline state of this material. So this Raman spectra just confirming with our conclusion in the DVS data that the sample will go through the amorphous to the crystalline phase change at 60% RH. So the combination of this Raman, the DVS, as well as the video images just give us a full idea about the solid characterization of a certain kind of material. So these just some basic information about the DVS technique, and then we will focus on some typical data. So this is the water sorption experimental data at 25 degrees for the microcrystalline cellulose material. And the blue line here is the target relative humidity at each stage, which is on the right-hand side here. And the right line is the change of mass for the sample, so it's on the right-hand side. And they are all changing as a function of time. So the water sorption data we perform here is from 0 to 90% RH with 10% different with 10% intervals. And at each stage, we will wait the sample to reach equilibrium. So for this microcrystalline cellulose, because the uptake is more than 10%, it has a very significant take-up of the water. So we just need to use a very small amount of sample, just around 6 milligrams, already get a very good quality of the water sorption data. So the total uptake for this microcrystalline cellulose is about 14%. And the total experimental time is about 2 days. And that's just the kinetics, sorption kinetics of this microcrystalline cellulose from the mass plot. And for the isotherm, is we taking the equilibrium mass change at each humidity, and we plot against the target relative humidity. Then we can get this isotherm with both sorption and desorption. So this isotherm will tell us lots of different information. So for example, at 90% RH, the total water capacity of this sample is about 14%. So the sample has very good affinity with water. And these water are not just sitting on the surface by the surface sorption, but it also goes to the bulk of the material. That's why there is a formation of the hysteresis gap between the sorption and desorption isotherm. So the gap here is called the hysteresis gap, is because of the water molecule when it's traveling into the bulk of the material, and during the desorption process, it's a diffusion limited process that will give a different kind of kinetics compared to the sorption. But at the end of the desorption part, it's still going back to 0, so it's a completely reversible process. So at the end of the process of this water sorption, all the water molecule will come out of the material. There is no structural changes at all for this abyssal or the macrocrystalline cellulose sample with water. So that's just the water sorption data at 25 degrees. And the case study here for the DVS data, we would like to investigate the temperature effect on the moisture kinetics of this microcrystalline cellulose sample. So we performed the same experiment as previously we introduced, from 0 to 95% RH with 10% intervals. And this time we performed experiments at a series of different temperatures. So from 5 to 60 degrees. So the amount of sample for all the experiments is just around 10 to 20 milligrams, because the macrocellulose sample has a quite significant uptake for water. And here are the results for the water sorption data at different temperature. So different colors correspond to different temperature, and between 5 degrees to 25 degrees here, the moisture uptake at a particular relative humidity doesn't show a significant difference. So for example, at 95% RH, which is the top point here, the water uptake for all different temperatures are all around 14 to 15%. But the time to reach equilibrium is much longer at lower temperature. So for example, at 5 degrees, the full sorption-desorption cycle will finish around 3 days. Well, for the 25 degree experiment, it just takes about 1.5 days. And also the equilibrium is not fully established at the last drying stage for the 5 degree run. So with the increasing temperature, the sorption kinetics of the macrocrystalline cellulose is much faster, and the equilibrium is also much better. Well, for the higher temperature, so for the 40, 50, and 60 degree run, with the increasing temperature, the water sorption capacity decreases a lot. This is probably due to the structural rearrangement of this microcrystalline cellulose sample. And also for the sorption kinetics, it keeps slightly increased with the elevated temperature. So the total experimental time for 60-degree run is just around 3 times faster compared to the 5-degree run of this microcrystalline cellulose sample. So that's just the temperature effect on the sorption kinetics of this MCC sample. And from the DVS data, we can also calculate the diffusion constant for the water molecule at different temperatures. The calculation of the diffusion for water is very similar to the diffusion calculation with thin film sample as introduced in the previous presentation. So we use exactly the same equation here, but in case of the thin film sample, we will use the thickness of this thin film sample, and in case of the powder sample, it will be replaced by the radius of these powder particles. So we have the mass at the time t and the mass at equilibrium state, and we also have the duration for the sample to reach equilibrium and the radius of the powder. Then we can get a fitting line of the curve, and from the slope, we can then calculate the diffusion constant of this powder sample at a certain RH conditions. So in case of the microcrystalline cellulose, the diffusion coefficient is calculated with the average particle size of 50 micrometers and at the stage from 70 to 80% RH. So these are the diffusion constants, also showing a trend of increasing with the increasing temperature, which means the temperature will help with the faster diffusion of the water molecule into the microcrystalline cellulose powder. So that's just some data about the diffusion constant at different temperatures. And another observation is about the hysteresis gap between the sorption and desorption isotherm. So here just a comparison of different temperature for the isotherm. The right line is for the 25 degree, the blue line is for the 50 degree, and the green line is for 60 degree. So as the temperature increases, the water molecule will move more freely out of the box structure, which results in a smaller hysteresis gap in this superimposed plot, and also an increase in the diffusion constant in the previous slide, because higher temperature has more energy input for the water molecule to go from the bulk to the surface for the desorption. So this just simple study demonstrates the effect of the temperature on the moisture sorption kinetics at a range of different relative humidity conditions within the temperature stability of the material. And we recently released the technical note for this study. So if you are interested, we are happy to share the notes with you after the workshop. So that's all about the DVS data. And the next technique I will introduce is the inverse gas chromatography. And this is more like a gas phase technique to mirror the gas-solid interactions to study the surface of the material. And the principle of IGC technique is rather simple, just being opposite of the traditional gas chromatography. So in the traditional GC experiment, we will have a certain kind of solid as the stationary phase. Then we will inject a mixture of the unknown compounds. And by the interaction, we can get 2 different peaks or multiple peaks here, just to separate the mixture being injected in the beginning. So from the retention time of different peaks, we can then get the information about the quality and quantity of this injected mixture in the front. And in case of the IGC, inverse gas chromatography, so this time we will pack the sample of interest into the column here. And we will do a single injection, a pulse injection of the single organic vapor through this column. So as a result, there will be 1 peak showing up here. So from the retention time and retention volume, we can then calculate the physical chemical property of the solid material inside the column here. This is a very widely used technique to measure especially the pharmaceutical material for different properties on the surface. So the procedure of performing an IGC experiment, we first need to pack the sample of interest into the column, like a glass column in the front here. And then we need to select different probe molecules for different interactions. So we need to select at least 3 different alkenes for the dispersive interaction and some polar solvent for the specific interactions. When we have the probe molecule, we can then inject them at different concentrations. And from all different peaks of the retention times, we can then calculate the physicochemical properties of the solid material we pack into the column here. So here are just some properties to be measured with IGC technique. So if we just do a single organic vapor injection at different concentration, then we can get the absorption isotherm of this organic vapor, for example, the octane isotherm. So from the isotherm, if it's a type 2 or type 4 isotherm, we can then calculate the BET surface area of this material. And if we are selecting different kind of solvents for different interaction, both the nonpolar probe and the polar probes, we can then get the dispersive and the specific surface energy of the material. And because we are doing the injection at different concentrations, so we can get a distribution of this surface energy on the surface, a heterogeneity mapping on the surface of this material. And from the surface energy, we can also calculate the work of cohesion and adhesion. And the balance between work of cohesion and adhesion is a very good parameter or indicator for the formulation study of the multiple component systems. And some other property we can also perform or measure with IGC. For example, we can get the Ka, Kb value from the acid-base interaction on the surface, as well as we can measure the phase transition for the glass transition temperature, or the Hansen solubility parameter with a series of different solvents interact with the solid material. But in this presentation, I just want to focus on the BET surface area of the microcrystalline cellulose sample. And the experiment we perform is just an octane isotherm at 30 degrees on the macrocellulose material. So here we use octane as the probe molecule. And according to the BET theory, if the shape of the isotherm is a type 2 or type 4 isotherm, which represents the monolayer formation mechanism, then we can use the points inside the BET range between 0.05 to 0.35 for the monolayer formation to calculate the BET surface area of the sample of interest. So here we have the relative partial pressure above the surface, and we also have the amount of the vapor absorbed on the surface. Then we can convert this isotherm using this BET equation here. This equation is exactly the same as the volumetric technique for the BET measurement as well. So with this equation, we can convert the isotherm into a BET line. And from the slope and the intercept of this line, we can then calculate the C constant, the sorption constant, as well as the Nm value, and that's for the monolayer formation. So from the monolayer volume and the cross-section area of the probe molecule we are using, in this case we are using octane, then we can get the BET surface area of the material we interested. So here we use octane to measure the macrocrystalline cellulose sample, and we pack 3 different columns, and each column we were measuring for 3 different times, just to get experiment-to-experiment reproducibility of the measurement, to get some confidence on the IGC determination. So the average value we measure for this macrocrystalline cellulose sample is just about 1 square meter per gram, a very low surface area. And as you can see, the relative standard deviation for each of the measurements is all below 2%, which shows a very good reproducibility of the IGC experiment, just as like chromatography technique, it will always give a very good repeatability. And that's the surface area just at dry condition. So it's only about 1 square meter per gram. And one of the advantages for the IGC technique is we have a background humidity controller inside the system that allow us to investigate the humidity effect on these surface properties. So we can perform very similar octane sorption experiments at a series of different humidity conditions. So here we performed from 0 to 90% RH. And as you can see, the surface area of this microcrystalline cellulose sample is quite constant and stable below 50%. So the surface area is just about 1.1 square meter per gram. However, when the humidity keeps increasing, the measured surface area will decrease, and at 90% RH, the value of the surface area is just around about maybe 85% of the initial value at dry condition. And the reduction in the surface area under high humidity environment could be correlated to the granulation of or the caking of the cellulose powder. So when the relative humidity in the environment is low, lower than 50 or 60%, the water content on the cellulose powder is also low, which resulting in minor extension of the granulation or the caking. But under high humidity environment, the water content increases, resulting in the significant caking effect. And this could lead to the lower BET surface area that can be measured with the IGC technique. So this simple case study just demonstrates how the IGC technique could help to study the humidity effect on the surface area of this MCC sample. We can also perform some similar experiment for the surface energy measurement to study the influence of the humidity on the surface energy as well. And that could be a further project for the investigation. And that's just for the case study about related to the microcrystalline cellulose I want to introduce today for DVS and IGC. And in case of the pharmaceutical industry, we do have lots of different applications with both of the technique. So for example, DVS can also use to determine the formation of hydrate, the kinetics of this formation, as well as the structure of this hydrate. As well as with some organic vapor, we can determine the amorphous content and the stability of the certain material at a certain condition. And with IGC technique is more widely used for the formulation design in the pharmaceutical material. So for example, the carrier and the API, the affinity with each other, and also the blending performance or behavior between different components in this system, or in case of the coating of the polymer on the base material, what's the affinity between each other, and that could be correlated with the surface energy value of each individual components. So that's the end of my presentation, and thank you very much for your attention. And if you have any questions regarding to the two techniques, just feel free to let me know, or you can also visit our website for further information, or just sending us email if you have any questions in the future. Thank you.
Thank you very much, Mei-Shan. And yes, a reminder to our audience to please submit your questions now for Mei-Shan to address, just on anything, any questions you have about that topic just discussed. Great, so we have a question here, Mei-Shan, and they ask, how would increased temperature affect the surface area of MCC at different relative humidities? Would the decrease in surface area be observed sooner, i.e. at lower humidities?
That's a good question. Actually, we are performing this experiment at the moment with DVS. So with the DVS, we can do the sorption of the octane isotherms. Then from the octane isotherm, we can determine the BET surface area. So probably the next study we will release is just about different temperature effect on the surface area of the cellulose sample. If we have the technician note ready, we will send you later maybe in the next presentation, or yeah, we will share the information with you.
Yeah, sounds like a webinar for 2022.
Yeah, that's right. And if you are interested in any of the topics we mentioned in our presentation, we are happy to share the presentation with you. And also we can send you some application notes on your request to any of the topic you're interested.
Okay, we'll just give everyone a minute or two more to submit any questions they may have. Thomas, before we finish up for the day, do you have anything you'd like to say to our audience?
Yes, I would like first to thank you, the University Mainz and Moritz, Rosch and Johannes for the clear presentation. Thanks to you. It's I would say benefit for all that you exchange the results with the audience you have. So thank you very much, and thank you to be guests. Unfortunately, still online, but I'm sure in the future it will be more on-site. If you give me the control, John, what I would like just to share to the audience is the way they can also. That's just a second. So if you would like to follow our series of webinar, you can go on our website, www.surface measurement systems, and you have here the tab webinars. And as you can see, there are a lot of webinars running.
We can't see the correct screen, Tom. We've just got your desktop.
I'm sorry, I lost, or I don't have control for some reasons.
It might be on the wrong screen, possibly.
Yes, perhaps. Okay, can you see my screen?
Yes.
Yes, okay, thank you. Yeah, so if you're on our website, you have on the top different section, and here webinars, and here you can see that's the today meeting, but you have all the past webinars we did, and here we announce also the future webinars. So don't hesitate to visit the website from time to time to look at the topics, if there are some topics interesting for you. Even in the past, if the reference have given their presentation free, there is some possibility to get a presentation over your sales manager. For Germany, Switzerland, and Austria, it would be me. The second thing I would like just to show you is we introduced last year different service contracts, and that is of course interesting for people having already an instrument. So an Exane contract, service contract, is, I would say, a maintenance as you know it. Yeah, but there are different levels of contract. Octane, for example, Octane it contains scientific and application support. I try to maximize that so you see that also. So scientific and application support. So that is very useful for people who want to get continuous application support from us. And what is coming now is also an e-learning portal, which is open for our customer having that, I would say, extended Octane contract. And okay, for the rest, you have multi-year and multi-system discount. And Decane is top of the line. So I would say here you have all included, all parts, all the travel, all the expenses. It's of course is more expensive than the Octane or Hexane. So Hexane is a perfect basic contract. Octane, a contract with application support, with more application support. Maybe also interesting if you have different people in the lab. Yeah, that's what I wanted to share with you.
Brilliant. Thank you, Thomas. So we don't have any more questions, so I think that brings us to the end of today's workshop. So a huge thank you to Johan, Moritz and Meishan for being here today to present on their recent findings, and also a massive thank you to you, Thomas, for organizing today's workshop.
Thank you. Thank you, John. Thank you, everybody. Have a nice afternoon.
Yeah, have a nice everyone. Yeah, have a nice afternoon, everyone. And if you do have any more questions about today's topics, sorption science or surface measurement systems in general, please don't hesitate to contact us at science@surfacemeasurementsystems.com. Or as Thomas has quite rightly pointed out, please do visit our website at www.surfacemeasurementsystems.com for more information. Thank you all and have a wonderful remainder of your day.
Thank you, everyone. Bye-bye.
Thank you. Bye-bye.
Thank you. Bye.